What? I sent a new email. Hi, James.
Be on him.
Y'all ain't time no take it's 5 o'clock.
Hi, Miss Doris.
Hi, Doug. How you doing?
Good.
I'm doing fine.
Did you get a ton of emails from me today?
Yes, ma'am. I got your email. Mm-hmm.
A ton of emails.
I got it. I think I saw it yesterday. Mm-hmm.
I see. No, these are today emails.
No, that's it.
Oh.
I sent it to Miss Jessie too. And I think Miss Lola, I'm not sure. I can never be sure with her email address.
Here's Lola right here.
Well, I think
you gave me on my secretary, Miss Doris.
Miss Lola, you're breaking up. I can't hear you.
She ain't saying nothing about you. Get put through her secretary me. don't pay her no mind. She done ate too much watermelon. Mm-hmm.
Oh, that's what it is. I saw them tomatoes.
Oh, Donna, they are so good. Oh my God, they are so good. Mm-hmm.
I'm telling you.
Now everybody wants.
Off on us now.
I only got 2 squash. I should have some more tomorrow. Mm-hmm.
Okay, you keeping track of them?
Yeah, you said weigh them.
Weigh them. Weigh everything that you get out of your garden.
I do, and I keep on track on how many pieces I get. You know.
Perfect.
Yep. Mm-hmm. I don't know what Jessica is. She must be. She running a little bit late. Mm-hmm.
Oh, it's not 10 yet. We got 10 whole minutes.
Yeah, I know. Mm-hmm. She had a busy schedule this weekend. She had family reunion at her house.
Wow. Yeah, that's busy.
Mm-hmm.
That is real busy.
All right, what y'all got down there? What y'all got down there to sell?
Huh. Listen, don't even ask at this point. We're suffering from, I lost all of my rice.
No.
Yep, because we planted too late and it was too hot.
What?
Burnt up. 24 beds of rice.
But you know, when I put that I can't think of the name, that spray in that white container on my tomato. I put some on my muscadine. Oh my God, I got so many muscadine on my vine.
Oh, that's sweet.
Oh my God. And then
that's the organics.
My plant, tomato plants, like they took a new look. They would look like they were dying. I gave them a shot of that, and they straightened up. They said straighten up and fly right. Mm-hmm.
That is fantastic. So which one is that? Is that the small one or is that the square 5-gallon bucket one?
Well, you know, at first I was using the square one. I was using I was using the 5-gallon one. Then I started change it up and use I change up.
Okay, so which one was it that gave the tomatoes a nice boost?
I got to go with the name. The one in the white container, the great big one. The great big one.
So that's that's the vermicompost. The vermi.
Yeah, the vermi extract. That one.
Oh yeah. All right.
But I did it by hand. I did it one by one. I gave my little bit of dose, one by one.
That's a foliar spray. That's right. Awesome. And then, and you said the grapes just bloom.
Man, they are so many. Yes, ma'am. Mm-hmm.
Awesome, awesome, awesome.
Yeah. Mm-hmm.
So we'll share that with everybody when they get online. And I'm going to do some tomorrow.
Oh, really? Oh, okay.
Mm-hmm. I'll use that one. We've been using the whole organics, but I'll use that one. Hello, Bill. Bill, you're
all right. We'll talk later.
Hi, James. Yes, Mr. Torres. That's all business right there, what you talking about. Hi, James. How you doing?
Dry. I need some of that rain that everybody else got. I didn't get a drop of that.
Australia.
Samantha just texted me and she said that her internet's out. She has this thunderstorm over her. She doesn't know if she's going to be able to get on. I told her, send me that rain. Just send it up here, you know?
We got rain in Massachusetts right now too. We've been very dry.
You've been very dry, but you're getting some. We got a couple showers, but I mean, we could use some more, right, James?
Yes, ma'am.
We could use some more. That's awesome.
The
Hi,
ground's
James.
cracking up here.
Oh, the soil's cracking?
Yes, ma'am.
Oh, James, I'll tell you one of the things that we're going to go over tonight. They suggested, NRCS suggested that I do another test that is going to look at the soil itself, like what the composition of the soil is. Like you have clay, some people have sand like me and stuff like that. So we will get that, we will be doing that in the fall.
Okay.
Yeah. So
I
that
did, I don't know if you'd recommend this or not, but on one of my beds, I put 3 inches of compost on a 30-inch bed, the whole 100 feet.
Hey, where did you get the compost?
That place in Texarkana for $10. It took about 1.5 yards, and it put about 3 or more inches on it.
Listen,
on the 30-inch bed.
That's fine because you're getting it at $10 a yard.
Yes, ma'am.
Perfect. And your soil needs it. So
Well,
that's excellent.
we'll see. I just wish I could get some rain. The sweet potatoes are surprisingly doing real well now, even though it hadn't rained. You know, I'm still watering it, but having that hay on there has made a big difference.
Yeah. Well, we're not going to have Conda on here tonight because she's in New York, I think, at Soulfire Farm. She's at a like a 1-week retreat on the farm, something like that. Anyway, so we won't have her, and I don't know if Ariel will be able to come on either, but they have changed their entire rice production based on the results of their patch with deep mulch with the hay. So yes, we're starting to see results. I mean, that was phenomenal because she has several other farmers that they're supposed to be meeting in August and coming up with a protocol for their farmers, which I'm one of them, that includes the deep mulch, the hay. So that's working out really well. So you're seeing that. And what she was saying is that even without the rain, when you pull that hay back, it's still moist under there. So it really makes a difference. But I'm telling you, where I don't have hay, I'm suffering. So we will be talking about cover crops on the field day. Remember, everybody, the field day is September 24th. Mark your calendars, and it's all day. I'm just telling you that right now. It's all day. Hi, Camilla, how are you doing?
Hi, Donna. We're doing good. I just now saw that you sent the soil results, so I'm looking into it.
Yeah, I said, well, they broke it out for me. And so I packaged them and sent them, but there are 2 that I sent everybody's. You'll get everybody's soil result on those. So you'll see that. You'll see the compaction test too. You remember when I did the penetrometer? Well, let's be honest. When Will did the penetrometer, Yeah, that is also, the results from that is also there.
All right.
Okay. Mr. Denton is on. Listen, y'all, you really don't want to see me tonight. I'm just going to tell you that. I am here,
Who's that?
but you don't want to see me, I promise you. I have been working all day, though. That, just take my word for it.
Hey, how you doing, man?
Hello, Anne. Oh, folks, I saw something that I have to make you aware of. So USDA, and Derek, you will be very happy about this. They are changing the way they're doing the insurance on specialty crops, and your crop plan and your records, planting records, can now be used. You don't have to bring in a third-party validator to go over your crop plan for specialty crop insurance. I don't know about commodity crops. I'm just going to tell you that right now. But for the specialty crop insurance, your records will be, you will be able to use your records, your planting records, your crop plan, and enter the program based on that. So I am going to ask the Farm Service Agency to come on. I'm not quite sure when. It may be on the field day, to just specifically talk about the specialty crop insurance, because Derek went down that road, and it was an interesting journey. And so now that they have changed the policy, I think it would be good to have them come on and talk a little bit more about that and what they're looking for. But basically, you don't need a third-party person to come in. You can just keep your records, and that will suffice. Well, of course, you're going to need photographs and all of that stuff, and there needs to be a natural disaster of some sort, but you know what I mean, okay? So that was a really good change for specialty crops. Who else is on? Hi, Josh. Okay, we're getting
Hey, everyone.
there. How's everything going, Josh?
Doing well. Summer school is finished for me, so I get to work on the house now.
Work on the house. What about ArcGIS, Josh?
That too.
Okay, okay. All right, so it's that time. It is 6 o'clock. Actually, it is 6:02, and we have a lot of special people with us today. So we have Bill Bean, who is our consultant. Hi, Bill. And we have Matthew Denton. And Matthew has popped in before to one of our classes. He is, Matthew, I don't know exactly what the right title is, but he is my direct report for this No-Till grant. And he is also in the soil health division. So I'm really excited that he's able to join us today. And he pulled in Dr. Josh Beniston, who is also with NRCS, to help us go through these soil test results and try to make some sense out of them, figure out where we are. This is our first one. And we've already discussed some of the changes that, some additional testing that would also be beneficial for all of you. So with that, I am going to, well, let me see. Dr. Beniston, are you ready? Maybe. I am on, I can see he is on. He has a picture up just like me. Both Anthony's are here. Samantha made it.
He was muted, Adonna.
Let me see if I can get him to unmute. Josh?
There you go.
Hello, how are you?
Yeah, hi everyone. It's Josh Beniston here from the NRCS Soil Health Division, and I'm doing well. Yeah, sorry, I just needed to step away a moment ago to fill up my water.
No problem at all. We were just, so you missed my brief introduction of you, but you can introduce yourself. They already know that you're with NRCS.
Okay. Yeah, so I'm a soil scientist with the NRCS Soil Health Division. And I work alongside Matthew Denton, who's a technical contact for your project. Matthew is our team leader for the Eastern US and my supervisor. But yeah, I'm here tonight to talk with you about soil data and some of the soil data that's getting generated in the project. And I suppose I'm here because I've been working with soil health data really for 15 years. I've been working as a soil scientist, my day job for the last 15 years. I've been a research scientist, both at the Ohio State University, as well as for the USDA, and also have been a teacher. I taught agriculture and soil science at Santa Rosa Junior College, which was a two-year community college in Northern California. And maybe of interest to all of you as well, that when I was there, I was part of a small farm training program, and also, worked on classes and projects related to growing vegetables and doing it regeneratively. And so we both tried to implement soil health building at a couple of different scales with vegetables. One scale was with the tractors. We had about 10 acres of vegetables that the team at the farm managed with tractors. And then my students and I had a garden that is kind of similar to how I picture the gardens that you all are working on. It was, we had, I guess 36 permanent raised beds where we didn't do any tillage or any serious tillage. We managed them with broad forks and compost and cover cropping. And the goal was to see how fast and how quickly we could build up soil health and have a system that was really productive and that people could do at small scales. A lot of my students didn't have a lot of resources. California land is not very accessible. You kind of take whatever you can get, and it's generally going to be something smaller, and maybe a partnership with someone else. And so we wanted, methods that people could use without a lot of land and without a lot of capital investment. And so we did a lot with small scale organic no-till. So, and I'm a big believer in small scale organic no-till. I think that's the way for vegetables as a soil health person. But yeah, tonight we're going to talk about the soil data that you all are generating as part of the project. And so, I would say there's a lot of it, right? We're kind of dropping you all squarely in the deep end here with the data. So this is a research project and you're generating a lot of soil health measurements for the project. But there's a lot there, and it's, what I've been looking at these numbers for, it's taken me 15 years to get where I'm at with understanding these numbers. So I would say, you know, we're all in different places with these measurements and numbers and view them differently and their value differently. But yeah, hoping that we can all learn some things together that will be useful to you all this evening.
So Josh, a couple things. I just made you host so that you'll be able to run the whiteboard and the meeting room. The
Okay.
other thing I definitely did the multiple presenter so that you can share your slides. But the other thing that I wanted to let everybody know is that the slides that you sent me was the third email that they got from me today. So if you go check your third email from me, then that's the one that he's using to present this afternoon.
Yeah, thank you for that reminder, Donna. And so, yeah, what that is I put together a little file that says CAMPTE example soil data for the class. And so what we're going to do with my portion of this is I have some slides I prepared that just kind of walk through what some of these measurements are and how we in the soil science world think about them. And then I thought we would use this this example data for some breakouts and discussion and trying to generate some questions so that you all can see the formatting of these reports that you're getting and some real numbers. And these are mostly from the Delaterre garden site that Donna is working with. And then I think we'll probably have plenty of time toward the end as well, that we can just kind of open up for all manner of question about these results and data that you're getting. And we, you know, happy to discuss any of the results that you're getting for your sites as well. And so I think with that, should I go ahead and get started on my presentation, Donna? Anything else?
Absolutely.
Okay.
Absolutely. Are you okay?
Yeah, I am. Sorry, it was just taking me a moment to navigate between. Yeah, at NRCS we were kind of all in on Microsoft Teams, so I haven't been on Zoom for a moment.
Listen, welcome to our world, and our world is very poor internet. So, you know, we have frozen frames and all sorts of gyrations occurring from time to time.
Okay, is everybody, are we good to people, folks?
That looks beautiful.
See my kind of opening slide here?
Yes.
Okay, so what I want to talk about, the outline for the talk tonight and the things that I want to go over is we'll talk a little bit about why we do soil health testing. And we'll start first with an intro to soil health and how we define soil health within the NRCS. And we'll look at the reasons why we collect these tests and how they may be useful to us. And then we're going to do a intro to some of these measurements that are being generated for the project. We're going to look at total organic carbon in soils. We're going to talk about permanganate oxidizable carbon. We will talk a little bit about water stable aggregation and soil structure. And then we'll look at the Haney soil health tests, probably the most the biggest chunk of data that we'll look at here this evening will be the Haney soil health test. So these are the ones that we'll focus on for the talk. Okay, so how does, well, actually, maybe before I launch into how NRCS defines soil health, if anyone else here on the call would would offer me some definitions or some characteristics of soil health, and you're welcome to put something in the chat here or to turn on your microphones. But what do you think? What does soil health mean?
Water
Anybody?
infiltration.
Yeah, water infiltration is something that we would like to achieve with healthier soils for sure. What else?
One thing we talked about in a previous class we had was resilience. So I guess, you know, the soil remains healthy as far as like water retention, nutrients, and biotic factors that, you know, even when the weather is not very forgiving.
Yeah, that's a great one. Resilience. I mean, I think that's a major motivation for why we're out here doing this work, is we want soils to be resilient, you know. I think where you all are at, you get too much water a lot of the time. Where I'm at, we have too little water a lot of the time. So we need soils that can perform well under both of those conditions. I see diversity of microorganisms. We'd like to see that for soil health as well. Absolutely, we would like to end erosion. That's you know, part of our story with NRCS and how we come to be here is a mission to end erosion in the country. So yeah, thank you for all of that. Those are all good points here for defining soil health. So within our organization, you know, where we you know, soil science is one of our focuses with NRCS. We have folks who are carrying out the soil survey, folks like Matthew and I who are out doing trainings around soil health, soil scientists. You know, we are one of the country's major organizations, maybe the major organization of soil scientists. And so, you know, we have our definition for our organization about what this term means. And for NRCS, we define soil health as the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans, right? So there's, you know, those are the key points. We need the soil to function. The soil is alive and we want to support its vitality. And we recognize that the health of the soil is tied to the health of agriculture as well as our own health and the health of our society. And so this is why we are concerned with soil health and what we hope to achieve. And so, you know, this is a look at soils and ecosystem services. And so what I mean here is that society derives a lot of services from nature and from natural ecosystems. And soils are at the center of many of those processes and services. And so some of the things that we depend on for soils, you know, we started my little discussion there started with someone mentioning water infiltration. So the hydrologic cycle, we need soils to provide plant nutrients. We need soils to break down all manner of organic wastes. We need them to support plant growth, right? Which is the business of agriculture. And we need them to moderate the biogeochemical cycles, things like the carbon cycle and CO2 fluxes from land. So, you know, we're depending upon soils for all of these services. And we know the research tells us that a different soil's ability to do these things is dictated by its health, right? This is the work of the soils that we depend on them for. And their ability to do that work is determined by their health, just like us, right? And what we have is a condition where we've allowed a lot of our soils to become sick. Many of them are underfed and overworked and stressed out. And so what we're trying to do is to correct that, to help them to provide these services for our benefit. So, here's how I first learned about soil health as a soil science student and researcher. I learned about it as a way of thinking about soils, and this is still, a big part of its value to me as a soil scientist. And so when we talk about soil health as a way of understanding soils, it integrates the chemistry of soils, right? That we want to have sufficient but not excess nutrients, and we want soils to be free of toxins. And chemistry, it's fair to say, was our focus for soils in agriculture for at least the second half, if not the majority of the 20th century. This is what we focused on, better soils through chemistry. And we made a lot of progress with producing food that way, but there were also some consequences, and there's also more to the soil than chemistry. I think that's ultimately what's most important to me about it. And so with soil health, we're taking a look at chemistry, but we're not stopping there. We're also going to look at the physical properties of the soil. We'll talk about soil structure for a few minutes with our wet aggregate stability measurements. We'd like to see soils support good rooting depth, like the picture on my screen. We'd like them to have good drainage and to resist degradation, as we discussed. And then the third major area that we want to look at is biology, right? I mentioned that soils are alive. And so we tend to be very interested in organic matter or carbon with soil health, as well as with populations of organisms in the soil, right? So we'd like to see beneficial organisms. We'd like to see small populations of pathogens and pests. And then, and so as a way of understanding soils, soil health integrates all of these areas, right? It puts them all together for us. And we understand that they are all interconnected. We understand that the overall health of the system, is dictating a lot about each of these three areas, and that we can't really just, it's important to not focus too much on any one of these areas. We want our management and our stewardship to address all of these. And so to me, this is what's powerful with soil health, is it's a paradigm about managing soils holistically. And then I'll just mention that photograph that's in the background there, that is from a soil pit in Kansas, in central Kansas, in a tall grass prairie. And so what you see on your screen there is approximately 6 feet from the surface of that soil to the bottom of that soil. And those plants, you see those plants, there's a perennial sunflower in the middle there that's shooting that big root down through the orange arrow, and a bunch of prairie grasses that are putting roots down. And so this is kind of the ideal condition. This is what we would love to achieve with our soils, right? This is a soil that's in an incredibly healthy and undisturbed state, right? And so it provides us a benchmark and a goal. And and this is the kind of thing that you all were kind of looking at with your fence row samples and your soil health data, is something like that, something that hasn't been disturbed as much. Whereas this is an intact native ecosystem that was never degraded, and so the soil reflects that here in the photo. Okay, and so those are our kind of concepts that we're working with. And then we're going to get down into what some of these tests are about. And so, why do we do this soil health testing? What can we achieve with this? And so you know, some of the things that we might achieve, we want to monitor changes in our soil over time. It's funny, this is my bullet points, you know, and so one thing we're also doing is determining a baseline. I think that's the data we have now for your project, right, as a baseline. What is the condition of the soil when we start? And then, you know, we can do follow-up testing and we can monitor changes. And with agriculture, usually what we're trying to do is to look at if our management has improved our soil properties. And that's what we're doing generally with soil testing at NRCS. We might also compare similar soils with different management or productivity, right? And so what I mean there is, if I have a farm or a garden and I'm doing something differently in one area in hopes of improving my soil, I would want to test that area separately. Right. So if I'm using a heavier compost application on half of my garden, I'm going to do it for a few years. I want to compare that. I want to see how that's changing differently from where I'm doing my standard management. And another way that's a really powerful tool for using soil tests is to compare areas of your sites that have different level of productivity, right? So what producers would often say is a good field versus a poor field, or a good area versus a problem area. And so, you know, oftentimes if we get testing done, we can see some differences between those areas. So productive areas versus problem areas. As I mentioned a moment ago, we might also look at farming areas versus natural areas, right? We tend to think that, and we have a lot of evidence that natural ecosystems tend to keep soil pretty healthy. They're very good for soil physical and biological properties. And so we'd like to use them as a benchmark. How are we doing compared to nature? And in our literature that we're developing with the Soil Health Division, we're saying that the primary goal of monitoring is to provide producers, managers, and planners with useful information so that they can optimize their given farm system, right? This is what we're hoping to get with a soil nutrient test, or with these soil health metrics that we're going to look at, is information that we can use to inform our management. And so some different ways that we can get at this, right? We're gonna focus on one of them tonight, but we often will use, I've done lots of soil fertility and nutrient testing in my background, as you know, a standard soil nutrient tests generally give you a lot of information about the chemistry in different areas in your soil, and generally things that you can do to correct things like pH or nutrient deficiencies. So that's one practice. Within our CS soil health, we are very big on in-field and visual monitoring of soil properties, right? This is something, I think Matthew is going to discuss this with you in the next webinar, is our in-field soil health assessment, right? Where we're essentially using a shovel and some simple tools and looking at what we can observe, just like the individual in that photograph there. So there's a lot about soil health that we can tell with our hands, with our eyes, with a shovel. Some people say good farmers can feel changes in soil health under their boots. I've experienced that in myself, right? It has to do with how hard the ground is or soft. So there's visual and tactile monitoring. We might look at production outcomes, right? So crop yields, right? This is how we kind of identify those areas that are doing well versus those that we know something's up and we're not sure what. Or nutrient use or water use if we have to irrigate, the use of inputs. We might also notice areas that don't, in California, we notice areas that don't dry out as quickly, right? And many places that you might notice areas that drain more quickly, things like that. And then what we'll focus on here this evening is soil health lab testing, which is kind of a new thing, really. So we're looking at these indicators of soil health from laboratory measurements. So in NRCS, we have a new activity that we are using with our producers that is just, I guess, just getting going this year. And the measurements that I have on the screen are kind of recommended list of measurements, and you all have an exciting project in that Donna has built in a lot of these measurements into your project. And it's exciting to us because for Matthew and I to get a look at a nice big set of these numbers that we're now expected to use with folks who want to look at their soil health. And so some of the lab tests, we're trying to look at the soil structural stability, which I'll talk about with you by measuring aggregate stability, which you all have in the project, as well as water infiltration. We're looking at organic matter cycling and carbon sequestration with soil organic carbon. We're looking at the carbon food source in the soil with permanganate oxidizable carbon. And then you all also have the Haney test that we have that we're going to look at tonight. Also has some measures of carbon food source. We are looking at microbial activity by soil respiration, which we'll look at a little bit tonight, and by soil enzyme activity, bioavailable nitrogen. We have this protein extraction that we're recommending at NRCS, but the Haney test that we're using in this project also has the bioavailable nitrogen test. And then the microbial community. So you all are going to get some data on PLFAs, phospholipid fatty acids. I think that's coming up for a future talk too. And as a scientist, that one's really exciting to me. You get a real detailed picture of the soil there. So these are the soil health lab tests that we're using within NRCS. And then we'll take a look at a few of them here. So one piece of data that you're getting for your project is the total organic carbon, right? And this is kind of the lab where I was trained to be a scientist. This was our focus. We focused on carbon, and I was really fortunate. I worked for a scientist named Ratan Lal, who's really one of the planet's authorities on soil carbon. And so for me, this is my personal sort of favorite. We're generating numbers with soil. This is my favorite one. This is the one I like to see. And so what this is the physical organic materials in the soil, right? It's a real thing. Total organic carbon is a real thing. It's anything in the soil that's made out of carbon. The lab will do a test where they heat it up to the point that everything is cooked and oxidizes, and they measure the CO2, and it tells them how much carbon was in that soil. And so these were materials, mostly it's from dead plants and dead microbes, the materials from plants and microbes, as well as their byproducts, but all living things. And we could measure it, right? It's a tangible thing, and we care about this because it's a driver, or it's important to most soil functions. The carbon tells us something about most functions in the soil. It's important for the structure of the soil, for the hydrology and water, both infiltrating and accepting water. Healthy soils filter water, and then retaining water for plants. All of those things can be predicted by how much carbon is in the soil. And then with microbes and the life in the soil, they need carbon for food, same as we do. And so it tells us, the potential of a soil to sustain life and supply nutrients. And it's also highly tied to the ideas we started with of resisting degradation and being resilient, right? We know we have just tons of data and research that tell us that soils with more carbon and more organic matter, they are more resilient. They resist erosion better, and they're more resilient to things like droughts and heavy storms. And so, for me, if I have to sort of do one test to give me an idea of a condition of the soil, this is the one. And so some ways to think about this. Well, this is a phrase we use with these lab tests, but more is better, right? We'd like to see more soil carbon. That's kind of, for me, again, that's kind of one of the main principles of regenerative agriculture, is we're trying to get more carbon into soils. And so, we want to see that number go up over time. And so my students, I used to have them call me Dr. B. And so my Dr. B's rules of thumb on total organic carbon, I think, what Dr. Lau, my mentor, what he taught us was that, after reviewing this across the world for 30 years, he felt like when your carbon number was below 1%, that was a soil that needed help and needed work, that it needed more carbon. The system was not at full health, was susceptible to degradation. And then here's how, this is a real tight window with this number. And then what I've noticed over my years is when we get carbon above 2%, then we really start to see noticeable improvements of soil health, right? That's something above 2% total carbon in a soil. That's a soil that's easier to put a shovel into. That's a soil that's going to infiltrate water better and hold on to water better. That's where you really see those things that we describe. It generally takes 3 to 5 years to make a big change in soil carbon. So it doesn't happen immediately, but it is hopeful in the sense that we can really impact this over the course of a life and a career and a project. And yeah, as I mentioned, for me, this is my number one indicator. This is my favorite for just, it's real. I know what it means. It's real. This is what we should be working on, and in my 2 cents on this. And so, I would make a note here. You may have, so there's a lot of terms around carbon and soils, right? We have both organic matter and organic carbon. So what's the difference? And scientifically, they're a little bit different, right? And so organic matter is that physical material that's out there in your soil, those pieces of dead plants and dead microbes, and it's made up of the element carbon. And it's approximately half carbon. On average, soil organic matter is 58% carbon. And so we generally just split the difference and call it half. And so what that means is something like, here's the other thing, is not all labs will measure total organic carbon. Donna did her homework and found a good lab for looking at soil health that provides you with this measurement. But a lot of commercial labs, they just focus on organic matter, which is still, useful data, but the numbers are different, right? So 2%, my 2% total organic carbon, that's equivalent to 4% soil organic matter, right? Or my 1%, that's equivalent to 2%, because they're a little bit different. When we say carbon, we mean carbon, right? And when we say organic matter, that's actual material that I could go out and hold in my hand in the field. And so, yeah, and we'll take a look at those reports here in just a few more minutes. So that's my sort of 2-slide summary on our total organic carbon. This is a big one. This is the one that can really lift all ships. And so it's good to monitor, and you can do it either way. You know, some of the labs we work with for commercial testing where I live, they don't offer this, so we use the soil organic matter test, and it's a pretty good proxy. But the industry gold standard is this total organic carbon, the test that has been done on your soils. Okay, one of the other indicators that we have from NRCS that's part of the project is this permanganate oxidizable carbon. And so what is this? This is the amount of carbon that is consumed in the laboratory oxidation test, right? They use this potassium permanganate chemical to consume the carbon. And the idea is that's a simulation of the microbes in the soil consuming carbon. And so it's considered to represent, so when I did a little bit of homework today to get ready for this, and reading the book, you know, it's actually about the microbe's ability to process carbon. You know, we often say it reflects the food for the microbes, but actually what it represents is their ability to process their food. And a lot of research studies have found that this is an early indicator of change in soils. It's kind of variable though. And so, you know, this is why we use it, and why it's recommended is because it can be used as an early indicator of change. And just like with total carbon, the idea is to have more, right? We'd like to see our permanganate carbon number going up over time. And that's kind of what we know about it. Gosh, I think I was just looking, like the original research studies that use this, the way that we use it now, were published in 2003, right? So it's a little less than 20 years we've been using this in the scientific community, and it's really taken off in the last probably 15 years. So that's not a real long time. And so, but what we do know about it is generally more is better. And you'll often hear people refer to this as active carbon, right? That's a little bit easier to say than permanganate oxidizable carbon. But, you know, the kind of uptight scientists said, you know, what the heck is active carbon? You should be calling this permanganate oxidizable carbon. So either way, but the idea here is that it is about the soil's ability to process carbon. And you see this picture I have here is from the Soil Science Society of America journal. And so that's a picture of someone doing the permanganate carbon test in a lab. When they take a soil sample and they digest it with the permanganate, it turns that kind of magenta purplish color. And they measure, that's actually what they measure, is the change in color of the solution. I forget which direction it is. I forget if more purple is more carbon processed or if a lighter color. But that's what it looks like, the active carbon test in practice. So, okay, last thing that I'll talk about before we take a break and look at some data and discuss is the wet aggregate stability. So this is another one that I like a lot, that the person who took this photo is actually a student who came behind me with Dr. Lau. But we did this test a lot. You know, a lot of the old school scientists in soils, they put a lot of value on aggregate stability. And so the reason is because this is the ability of soil aggregates to resist breaking apart in water, right? So this picture is a close-up photo of soil aggregates, and they're sitting on a wire mesh sieve, if you can picture that. And that sieve gets dunked up and down in water for a set period of time. And then what wet aggregate stability measures is what percentage of your soils held together when they got dunked up and down in the water for a set amount of time. And there's a bunch of different ways that people do this, but that's the basic principle, is taking the soil aggregates and subjecting them to repeated dunking on top of the screen, and what stays on the screen. And so the idea is that this is an indication of how well the soil holds up, right? Its ability to resist disturbance, right? And so when your soil holds up like these aggregates that stayed together, that's a good sign. That translates well. That's why the older generation of soil scientists like this, is that translates to the field. That same soil that holds up well on that sieve is going to hold up under 3 inches of rain out in the field. It's not going to wash away and go anywhere. It's going to stand strong. And so that's what this is about. As you know, a way that we came up with to try to understand that in the lab. And so again, more is better here. I took a look at all the numbers from your project, and I thought they looked quite good overall. You all have decent soil structure across your project. We tend to think somewhere in the 50 to 60% of your soil having aggregate stability is a good sign, and that was the case with most of your samples. So I gotta have to go back and fix that my little first points coming up last, but that is the aggregate stability. And so I don't know what everyone's background is on this, but I put this in there again, and so part of why we care about this aggregate stability as well is because we have this term structure, right? Structure in soils means small particles sticking together into larger particles. And so how do they stick together? Usually because of the action of living things, which this photograph or this drawing demonstrates, right? That there's these big white tentacles on here. Could be fungal hyphae is what it's labeled as, or it could be small roots. And so basically, the biology go out there and they physically compress the small particles in the soil. And they also are exuding things that are sticky to help the soil stick together. And as they do that, and as the small particles stick together, it opens up pores and space in the soil. And that is so important because that's the space for air and the space for water and the space where living things who need to breathe oxygen live, right? And so, and you can't have one without the other, right? And so this process of aggregation, what it results in is more space, more open space in the soil. And that's good for plants and it's good for microbes, right? And because it's space for air and it's space for water that they need. And so, this is a major thing for us in soil science. We're looking at aggregate stability and the density of the soil because we want to see open space for roots, right? Because they actually, all these living things, they have to live in open space, and the plants, they have to physically push their way through. And so if they can take advantage of open space in the soil, that's really advantageous for them. Okay, so I think from here, we're going to discuss some data for a little bit, but are there any questions? I just kind of threw a bunch of information at you in the last half an hour here. And so are there questions on what I went through so far before we move on? Any questions? Okay, well, what we're going to do in a moment then is we're going to take a look at this file here. So this is our this example data. And so, this is me opening the file on my computer. So if you can get this file out, because I'm going to ask you to take a look at it and provide me with some reactions. And so, this file here's got 5 pages. And so I believe the first page has the organic carbon, right? These are the samples from the Delaterre site. And they give you the carbon number. Down here in the lower column, there are two numbers. And you can see the farm plot is the top row and the fence line is the bottom row. And then, you know, this other page, the next two pages have everyone's numbers. We're going to look at the Delaterra data, but you may want to look at your own numbers as well. And this has got the permanganate carbon data here in the middle. And then over on the far side, it's got the wet aggregate stability. And so with aggregates, we also break them out by their size. So we have macro aggregates, which are larger, and micro aggregates, which are smaller, and then the total number. And then so we're thinking about the total number, and then, you know, what I'd say as a general rule, you know, a kind of a generalization, we like the macro, the bigger aggregates for soil health. So we're going to look at these first 3 pages. And then if I scroll through we'll come back after this and we'll talk about these Haney tests, which have a whole bunch of information right here. But to start with, we'll talk about total carbon, page 1, and then permanganate carbon and aggregate stability on page 2 and 3. And so again, this is this CAMP-T example soil data for class file that Donna sent out. I think she said in her 3rd message to you all today. So if you can open this up, what we're going to do is I'm going to force you to talk to other people now. I'm gonna drop everyone into a breakouts here in a moment, and then I will have some questions. So let's see. We'll go, yeah, we're gonna have 5 rooms, 3 or 4 participants. So you'll go in there with a group of other folks. And what I want us to think about here, let's see here. So again, before I do that, any questions about the stuff that I went over?
And should I give any background to that, to the samples, or?
Sure, absolutely. Yes, if you think that if that would be helpful, then please. Yep.
So the fence line, so this is very sandy soil. And I was actually really impressed in the total aggregates and the amount of aggregation, because when you look at the organic matter on the fence line, it's 0.6, not the organic matter, but the total carbon, it's 0.65. And in the plot, it's 1.28. Well, we've been adding compost and things like that to the plot. The fence line is really low, but look at the amount of macro aggregates on the fence line. So I thought that was very interesting. Yes, higher is better, Wellen.
Yeah, higher is better. Yeah, so I think let's go ahead and take a look at these. So let me see here. I have a few questions for you all. Let me bring this back up. So in a moment, I'm going to open up these breakout rooms and you're going to have to click to join. And then I think we're going to take, let me take about 6 or so minutes. And I'd like you to discuss, I'd like you to take a look at this file and I'd like you to discuss with the other folks that you're in there with. And so what I'd like you to discuss are the following points. Take a look at that Delaterra data. You can also take a look at your own data once you're in there, but just kind of what are your observations? What do you think about the measurements we looked at? So again, what we want to look at now is going to be the total organic carbon, the permanganate, and the wet aggregate stability. Is there something you find interesting in those 3 numbers? Is there something that's confusing? So that's part of why I'm here tonight, is that this stuff is confusing. So if we have questions, please bring some questions forward. And then, you know, if you want to scroll down and find your own garden site in that permanganate and wet aggregate stability data, how's it look based on what we discussed? What do you think? How's it look for your site versus the Delaware site? So those are the questions, sort of. What are your, and I'll stick those in the chat if I can here so that we don't forget. That's what I'd like you to do. So I'm going to stop sharing here. I'm going to open these breakout rooms. So I'd like you to join your room. And then these are my questions.